Now the biochemical changes. The biochemical changes which are induced in the virus infected cell. What is happening when the virus is infecting a host, and then what are the biochemical changes which are implementing inside that host, that how the virus is going to replicate and taking the charge of the host machinery, what are the biochemical changes which are happening inside the cell. First of all, the basic question is it that virus induced changes would be able to induce the cell death, or they will just only accelerate their growth. Sometimes the virus which is infecting the host, it is just replicating itself, it is not causing any damage to the host. But most of the time the virus is taking the charge of the machinery and taking the charge of the host whole machinery for its replication. It is damaging the host, which can cause the cell death. Answers will be hidden in the different proteins which are encoded by the viruses, and the proteins depends on the cell death, or they are just going to help in its own replication. So we will see it. What is the virus? For example, we take the example of the picornavirus. Picornavirus are the smallest viruses. However, even being the smallest viruses, they are containing 4,000 to 6,000 nucleotides inside their genome. Even the smallest viruses on the earth has the genome of 4,000 to 6,000 nucleotides, and genetic information codes from the 1,500 to 2,000 amino acids, and total proteins which are about 10 to 15, of course, which is composed. Therefore, this is the smallest viruses on the earth, and how many proteins are being encoded by them, and then the total amino acids are 1,500 to 2,000. Similarly, there is the vaccinia virus. The picornaviruses are considered the smallest viruses. Similarly, the vaccinia viruses are the big viruses, and they are containing about more than 2 lakhs nucleotide pairs in between them, and which are encoding about 400 to 600 proteins. So therefore, from this comparison, you can compare the two different kinds of the viruses. One virus is causing about 10 to 15 proteins encoded, and the other virus is encoding 400 to 600 proteins. And the specific virus and the cell death, the virus which is infecting to that specific kind of the host, depends on the proteins, which kind of proteins, and how much is the amount of the viruses which has infected that specific kind of the cells. So there are two different kind of the proteins which are infecting the disease. One is the early proteins, and other are the late proteins. Both of the proteins have some different abilities or some different properties. First of all, we see the early protein synthesis usually happens due to genetic function of the parental viral genomes. Because of the presence of genetic viral genomes, the early proteins synthesized, and their function includes the inhibit the host cellular DNA replication. This host cellular DNA replication occurs in the early proteins, and similarly there are some also other functions just like the host cell RNA formation and host cell protein synthesis are also occur because of these early proteins. While on the other hand there are late proteins which include the viral structural proteins. The structural proteins are very important. Of course, we have already studied the structural proteins and non-structural proteins. And these proteins are the structural proteins, and these functions in suppression are switching off the formation of virus, and also switching off the viral enzymes, which are also playing very important role with the biochemical changes which are induced inside the cell.